Welcome everyone to this week's edition of the Extracellular Vesicle Club. This event of the International Society for EVs has been running for almost 2 years now, and my name is Kenneth Witwer. I'm the Chair of Science and Meetings for ISEV, and I've been hosting this event. It is my pleasure to introduce our speaker for today, Oskar Staufer. So he's done a lot of work in Heidelberg in Germany, and he is now a postdoc in Michael Dustin's lab at University of Oxford in the UK. So Oskar has been working on something that I think a lot of us think about, because when we contemplate the therapeutic uses of extracellular vesicles, we know that there is a certain amount of heterogeneity to these vesicles. And so the one vesicle that might contribute to neurodegeneration might be next to a vesicle that is very different that fights neurodegeneration, and so on, in whatever model you're working with. So this diversity of extracellular vesicles is a strength, but also a potential weakness. And so Oskar has been working on how you might actually assemble an extracellular vesicle completely synthetically. So I know that we're all looking forward to learning more. Oskar, thank you so much for joining us today, and I'm going to hand the screen sharing over to you.
Thank you very much. Okay, hello everyone. As Ken said, my name is Oskar Staufer. I used to work in the Department for Cellular Biophysics at the Max Planck Institute for Medical Research in Heidelberg. That's also where most of this work has been performed in collaboration with people from the Leibniz Institute in Aachen, also in Germany. And the paper I will be presenting to you today has been published in Science Advances about 3 months ago now. I hope this will be an interesting hour or so of discussion and presentation with you guys, the experts in the field, because actually I need to start with a little confession to this, which is I kind of came into the EV field from a very different perspective, a field that's not naturally related to extracellular vesicles, and that field is bottom-up synthetic biology. And while this is a diverse audience that's very much working on extracellular vesicles, I think bottom-up synthetic biology deserves a brief introduction in this context, which is why I will dedicate one single slide to introduce you to the mindset of this field, because it has been very important for the research that I will be presenting. So bottom-up synthetic biology in general is a field that strives to construct biomaterials that mimic lifelike components in a very minimalistic way. So basically, in this field, we try to reconstitute cellular phenomena in vitro. Well, we do this because we know as biologists that living systems are very complex, from the molecular level, well, down at the molecular level, up to the interactions of organisms. So if we look at a simple but very important structure for life, which is the ribosome, we can appreciate that those systems already are very dynamic in their complexity. So in bottom-up synthetic biology, we try to construct systems in a very isolated environment that we can control down to the quantitative level in order to extract quantitative information from those systems, and then understand in a more systematic way how these individual components work and act together in living matter. Obviously, we tackle some very profound and interesting questions in the life sciences, which is, where is the actual transition from matter in living matter to living matter? How can we integrate synthetic systems into living materials such as cells? And that is also a point that I will be tackling in the later part of this presentation. How can we actually engineer lifelike systems on a quantitative level, for instance, for biomedical applications? So one thing that does not need too much of an introduction in here is extracellular vesicles themselves. I just want to make sure what I understand in terms of extracellular vesicles and how this term will be used in this presentation. Extracellular vesicles are a diverse set of vesicles, so lipid enclosed structures released by cells, either from the intracellular environment or from the plasma membrane of cells directly. They carry bioactive molecular cargoes, which could be RNA, proteins, DNA, sugar, or whatever else. And we know from extensive decades of research now that these structures are very complex, at least in the molecular composition. We have identified thousands of proteins and RNAs that can be found in extracellular vesicles. And what might be even more important for the therapeutic spectrum of these vesicles is their functional diversity. extracellular vesicles have been pinpointed as very important structures in cellular communication in a diverse set of cellular processes that might range from antigen presentation to bone resorption, but also inflammatory processes and so on. Essentially, I think, at least for human biology, there's barely one process that has not been connected to extracellular vesicle signaling so far. Yeah, so obviously extracellular vesicles are important for our work, and if we want to understand in a holistic way how cells communicate with each other. That's not only interesting from a fundamental science point of view, but it's also very interesting if we want to apply these structures for biomedical applications. Why we want to do so? Well, obviously EV-based diagnostics have been very important in the last years of research, because we know that extracellular vesicles are somewhat mirroring the physiological states of their cells of origin, and we can somewhat easily retrieve them from biofluids of patients. But what might be even more interesting is that we can reapply or re-administer purified extracellular vesicles, either from patients themselves or produced in cell culture systems, back to patients in order to trigger and make use of the endogenous regulatory functions of extracellular vesicles. Well, we have done a lot of work on this as a scientific community, but we are still somewhat limited, as Ken said in the introduction, by some systematic limitations when it comes to extracellular vesicle research. And that might be that the production conditions are fluctuating a lot. So basically, you can get two very different sets of EV populations from a cell culture, depending on how dense the cells are that are growing that cell culture, not to speak about serum composition, types of cells, and so on. Another limitation is it's very time consuming, that process, especially if we think about industrial scales, where we need to scale up the production from one petri dish to several liters of culture medium. But the whole process of EV production, isolation, and purification is somewhat error prone and unstandardized at this point. And I think the technique that I will be presenting to you today and what has been published in that paper at least can help at some points in these limitations to get a step closer towards a biomedical application of extracellular vesicles, but also of a deeper understanding of their signaling mechanisms. So I think the major problem that underline these limitations is the complexity and the diversity of extracellular vesicles, as I introduced in the beginning. That has mostly limited our quantitative understanding of the signaling behind extracellular vesicle communication. It has impaired a programmable engineering of those structures in a quantitative way, which is why we have mostly been able to engineer extracellular vesicles in a top-down designing way. So basically genetic engineering of their cells of origin, or any kind of biological chemistry that we can apply to extracellular vesicle purifications in order to tune their physical, chemical, or biological properties. So what I will be introducing to you today is a complete new approach to extracellular vesicle research, which is aimed towards the bottom-up assembly of fully synthetic extracellular vesicles, so structures we call FSEVs. And that is essentially applying the mindset of bottom-up synthetic biology for the construction and study of fully synthetic extracellular vesicles in order to explore the therapeutic potential of such systems, but also to analyze the functional signaling mechanisms behind extracellular vesicles. So none of the vesicular structures that you will see today are actually produced as depicted on the left side of this image in cell cultures or patients, and they have not been isolated, and there's no natural vesicle population that is poorly defined either in the physical, chemical, or biological point of view. But all the vesicles I will be showing to you today are actually produced starting from the very molecular components, that is individual lipids, proteins, and RNAs. And I will be showing you how we can apply a quite simple, actually, but chemically complex in vitro assembling technique to create a very defined population of vesicles that mirror extracellular vesicles. And I will show you how we can apply these fully synthetic constructs for therapeutic purpose, along with studying the signaling mechanisms of these vesicles in a very systematic way. Before doing so, I will need to introduce to you how we actually assemble that process that's shown here on the right, the in vitro assembling technique. That's a technique that has been developed in the Department of Cellular Biophysics at the Max Planck Institute for Medical Research a while ago for the production of synthetic cells, and that has been reapplied in this paper for the production of extracellular vesicles, basically lipid enclosed structures that are much smaller compared to living cells. I also want to say upfront from this slide, if I don't manage to explain completely to you in detail and comprehensively how this process is done, it is not absolutely pivotal to understand it for the upcoming slides. Essentially, the important thing to understand is the outcome, which is depicted on the right side, which is a very well-defined vesicle population with proteins and nucleic acids like RNAs in the lumen. So let me start introducing to you how we assemble these fully synthetic extracellular vesicles. Essentially, this is based on a water and oil droplet microfluidic system, although the microfluidic part can be neglected in this stage. And it starts by interfacing an aqueous layer that could be PBS with an oil layer. In that case, the aqueous layer contains the RNAs, for instance, in this project, microRNAs that should land up in the lumen of the fully synthetic extracellular vesicles, and the lipids that are in this case provided by liposomes that should form the membrane of the fully synthetic extracellular vesicles. While the oil layer of that interface will consist of charged surfactant and bioinert perfluorinated oil. These two layers are emulsified either by mechanical emulsification or by a microfluidic approach. And this is how water and oil droplets are formed that are surrounded by a charged surfactant polymer layer. And this charge of the surfactant layer will actually lead to the fusion of the individual lipids provided in the initial solution with the periphery of these droplets, incorporating the nucleic acids that were added to the PBS or the aqueous layer initially. So this is basically a shell-protected, fully synthetic extracellular vesicle that we now need to get out of this protecting and assembling polymer shell. This is simply done by destabilizing such an emulsification back to a phase-separated system, where we have then individual vesicles with the size, depicting the size of the droplets that were initially formed, and with microRNAs inside. The only thing we need to do now is actually couple proteins to the surface, ectodomains of proteins to the surface of these vesicles. That could be done either by NTA, nickel, histag chemistry, by biotin, avidine linking, by covalent linking methods like NHS chemistry whatsoever. But we also have developed methods how to incorporate full-length transmembrane proteins into such lipid structures. So this is the essential process of the formation of fully synthetic extracellular vesicles. And the nice thing about this is actually it can be performed within one epi, as I'm showing you here in that graph, simple by manual shaking of that Eppendorf tube and producing water and oil droplets. So there's no microfluidic system needed for this, and it can be applied by probably in every lab with standard lab equipment like centrifuges and pipettes. So what are the structures? How do they look after the formation process in the real world? What I'm showing you here now is a confocal image slice of such a vesicle population. You can see that we have a lipid enclosed membrane. We can tag typical EV proteins like CD9 ectodomains on their surface. We can incorporate microRNAs into the lumen. Essentially, the advantage of this is not only on the molecular complexity level, but also that we will get the more defined vesicle subsets. This is a dynamic light scattering analysis of a typical natural EV population. You will see different sizes in this, for instance. But when you analyze fully synthetic extracellular vesicles, the size distribution is actually way more narrow and way more defined. That can be a very systematic advantage when looking to towards the therapeutic applications of extracellular vesicle biology. Most importantly, however, fully synthetic extracellular vesicles are internalized into cells and are directed into the endosomal compartment of these cells, as is shown in this confocal set stack movie through, in this case, keratinocytes, human keratinocytes. On the left side, you can see in yellow the fully synthetic extracellular vesicles, and on the right, the endosomal compartments. So having this technique in hand, what do we do now? Essentially, we apply our bottom-up synthetic biology mindset and look what nature gives us in terms of a blueprint that we can start to reconstruct in vitro. Essentially, we started our study by looking at a paper that was published by Geiger and colleagues a while ago, and that describes human fibrocyte-derived exosomes that accelerate wound healing in a diabetic mice model. So Geiger and colleagues are part of a company that was based in Berlin that has now been sold to a Canadian company that was trying to establish fibrocyte-derived extracellular vesicles for wound healing purposes. And what they did very nicely in their paper is they characterized the phenotypic changes that these fibrocyte-derived exosomes will induce in different cell culture systems. For instance, they found that when incubated with epithelial cells, they will induce a pro-proliferative effect, as well as an increased collective cell migration of these epithelial cells. Similarly, when they incubated these exosomes with collagen-producing fibroblasts, they could show that there's an increased collagen deposition by these fibroblasts. All of these three processes are obviously very important for wound healing processes. Most importantly for us, they also performed a molecular analysis of the components that are found in these extracellular vesicle populations, that is, the detection of typical EV markers like CD9, 63, and 81, but also some microRNA components and some typical lipids that we will usually find in extracellular vesicles. So we used the paper published by Geiger and colleagues as a blueprint to construct fully synthetic extracellular vesicles, and then assess if we can employ the synthetic structures that we created for a wound healing therapeutic purpose, but also to decipher in a systematic way which of the individual components are actually important for the signaling and the therapeutic effect behind these structures. So we started off by measuring one of the simplest thing is actually proliferation of cells in culture after incubation with our fully synthetic extracellular vesicles. And what you can see in this quantification of cells in culture after 48 hours of treatment with our fully synthetic extracellular vesicles is that we can not only prove that they somehow increase the proliferation of cell numbers in this experiment, but we can actually check each component one by one and its effect on this process, either one by one or in combination of the different proteins. That is something that's very hard or maybe even impossible to do with natural extracellular vesicles. We can not only do this with proteins, we can also do it with the microRNA components. And essentially what we found is that the structures we produce will increase the proliferation of human cells. And we're also able to systematically study in a sequential, functional way, which are the EV constituents that are important for this pro-proliferative effect. The same thing we can do now in order to analyze the increased cell migration of human keratinocyte cultures in vitro. That is essentially done by in vitro scratch assays. The experimental procedure behind this is quite simple. Basically, one grows a cell layer of keratinocytes to full confluency and then introduces an artificial wound site by scratching, for instance, and then simply one follows by microscopy the closure of that artificial wound site. That can be quantified over time or either in an endpoint analysis, like in this quantification here. And basically, we did the same thing as reported in our paper. We measured the impact of individual protein or RNA constituents on the closure of that artificial wound site. And again, we could not only find that this artificial wound sites are closed more rapidly when we incubate cells and keratinocytes with our fully synthetic extracellular vesicles, but we can also again show which are the important constituents that actually have a very increased effect in that phenotypic transition. Well, essentially, we did the same for the collagen cultures. I'm not showing this now here in the sake of time, but essentially, we could also show that the full synthetic extracellular vesicles increase collagen deposition by human dermal fibroblasts. What I want to show you, however, is that we can now apply the system in a more physiologically relevant scenario, that is in human organotypic skin models. Those are skin models that have a dermis as well as an epidermal layer, and that can be wounded very specifically, for instance, here in the epithelial layer. And the closure of a wound site over time can be followed by histological staining afterwards. So I'm showing you here the initial wound site of 3 millimeters. That would be an initial diabetic ulcer. We can also then increase the wound healing with human serum that will contain natural extracellular vesicles, or either with the fully synthetic EVs that we apply directly on the wound here. And when we quantify either the wound diameter from those histological sections or the epithelial thickness of the regenerated epithelium, we can again see that fully synthetic extracellular vesicles can increase these processes and be beneficial for the healing of such human organotypic wounds. So essentially, the fully synthetic extracellular vesicles that we constructed based on the natural blueprint are actually of comparable therapeutic functionality as their natural blueprints. And that is, I think, a very important thing to show, that we also did in the paper, that we can use EVs not as therapeutic entities themselves, but rather as an inspiration to construct synthetic biomaterials that mimic their properties for a therapeutic benefit. What I want to show you as well, however, is that this can not only be done in order to engineer new therapeutic approaches, but actually also to dig a little bit deeper into the signaling mechanisms behind extracellular vesicles. And this we did simply by coupling our bottom-up synthetic biology approach with a technique that is rather used by top-down researchers, which is a simple transcriptomic analysis of the changes in the gene expression signatures of keratinocytes that were treated with fully synthetic extracellular vesicles for 24 hours. So essentially, we found several differentially expressed genes in these cultures. We could then, by pathway enrichment analysis, pinpoint which are the pathways triggered by the treatment with the fully synthetic extracellular vesicles. Those were, for instance, as marked here in orange, pathways that are somehow important for cell proliferation and cell cycle progression. So obviously things that are behind the pro-proliferative effect of these structures, but also all kinds of pathways involved in cell migration, cell adhesion, as well as extracellular matrix remodeling. This is an analysis that could be easily and probably more accurately be done with natural extracellular vesicles. So again, I want to show you how transcriptomic analysis can be coupled with the bottom-up approach by now testing the pathway, by now applying the pathway enrichment analysis with single fully synthetic extracellular vesicles that show single, in this case, protein constituents on their surface. And now we can resolve actually for each pathway, which are the important protein constituents of such a vesicle in order to trigger individual effects. And I just want to point out one very interesting combination that would be a combination of CD9 and CD81, that this alone, while the individual components alone do not trigger immune-related signaling in the keratinocytes, which is obviously an important process in wound healing in order to prevent the entry of infectious agents. The same thing we present in the paper for the microRNA system. So you can disentangle and decipher the contribution of individual components in natural extracellular vesicles to a phenotypic and maybe therapeutic effect based on the signaling mechanisms behind this. The last thing I would like to show now is that nature can be seen as a blueprint in order to construct minimalistic biomaterials, but it can also be used, so this technique can also be used to go some steps beyond nature. So we can now also engineer and optimize the fully synthetic extracellular vesicles in order to perform best for the phenotypic change that we actually want to use. In this case, I'm again showing you the migration data for in vitro analysis of collective cell migration. And what we tested now as an example is actually what happens if we systematically increase one of the microRNAs that has been pinpointed in the previous analysis as very important to induce this migratory effect, which is microRNA 132. And if we vary the ratio between this RNA and the other RNAs, and what we can actually see is that there's a kind of an optimum reached at a specific concentration. And if we increase that pro-migratory RNA even further, we actually lose effect again. The same thing we can do for protein components. In this case, we vary the surface density of CD81. And we can actually show that in this case, there's rather cutoff behavior where at a specific ratio, the migratory behavior of the cells increases at some point, and further increasing the density of CD81 on the surface is actually not beneficial for the therapeutic effect that we want to induce. So these are some exemplary results that should just highlight that fully synthetic extracellular vesicles can also be engineered in order to optimize a specific therapeutic effect that we want to induce. And that is something that I think has been started to be done in natural extracellular vesicles, but that can be done here in a way more quantitative way at the end. So let me conclude what I've been showing to you today. First of all, I hope I convinced you that we provide a first proof of concept that we can design and assemble some kind of programmable synthetic extracellular vesicles that mimic, in a certain extent, the functionality, at least in a therapeutic way, of extracellular vesicles derived from natural cells. I showed you, and that's also highlighted in the paper, the identification of key microRNA and protein components in that system, especially on a quantitative level. And eventually that fully synthetic extracellular vesicles and the technology behind this can be applied for a very systematic analysis and thereby overcome the systematic limitations of extracellular vesicles by breaking down their complexity and diversity. So just a brief outlook into the future. Well, obviously, we think that this technology has the potential to bring forward a very new class of therapeutics that are based on a bio-inspired system and merge synthetic and lifelike materials. We hope to apply these systems to create synthetic communicative environments for cells, either for research or for therapy. But I think one of the most important points of this technology is it can be used as a standard for experimental procedures that focus on natural extracellular vesicles. I think in many EV production, purification, and isolation procedures, standards are needed that can be added as an internal standard into any kind of a sample in order to quantify and quality control purification, isolation processes. And this is one of the lines where we are driving this technology to, in order to provide hopefully a new valuable technology for the whole extracellular vesicle research field. Let me close by thanking the people that were important for the study. First of all, Joachim Spatz and Ilya Platzmann, who have been supervising and directing this research, as well as obviously all the co-authors on the paper. Many collaborators, either at the Max Planck Institute for Medical Research. I just want to highlight Franziska Dietrich, who is second author on this paper and has been very important in order to perform the organotypic skin model work, as well as Rahul Rimal from the Leibniz Institute in Aachen, who has been very important in analysis that I could not show to you today, which concern the induction of neoangiogenic processes by the fully synthetic extracellular vesicles. And I would like to thank you for your attention, especially as I did not do this in the beginning. I would like to thank Ken for inviting me and for exposing me to the extracellular vesicle community in this format. And I'm happy to take questions.
I am now going to allow unmuting. So if you have asked a question, please be ready to turn your microphone on and interact with Oskar here. So let's start out with Miguel Silva. Miguel, do you want to go ahead and ask your question about combining things?
Hello, I'm at a lab right now, but my question would be, what is your opinion on bridging natural vesicles and synthetic bottom-up approaches into a top-down approach, just like cell membrane-coated nanoparticles or cell membrane-based nanovesicles? Thank you very much.
Thank you very much for that question. So I think this is maybe also a very interesting application of the technology itself. Obviously, if we are able to merge natural vesicles with synthetic components, for instance, synthetic membrane components, that could be helpful if we would like to engineer standards for the purification processes, but it could also help to introduce rather synthetic functionalities into natural EV therapeutics that would expand the therapeutic spectrum of those structures. So I think there's a lot of potential that has not been met yet, although there are many approaches that start to research in that direction. But I think the next 10 years would be very interesting to follow up such a technology, because I think the techniques we need in order to create such structures are there, and now we just need to look at how well can we engineer them and where can we apply them.
Thank you. Thank you.
Thank you. Next question is from Ruben Lopez Salazar. And Ruben, feel free to jump in here for any follow-up, but he asks, how do you form the initial SUVs?
So these liposomes, so small unilamellar vesicles that we apply, are simply formed by standard extrusion through nanoporous membranes in that case. So that's a quite simple technique, although it is not limited to these kind of vesicles. Yeah, we can use all kind of liposomal to vesicle structures in order to produce such fully synthetic extracellular vesicles. Obviously, what you would like to make sure is that this is a unilamellar system. So we only have one lipid layer around, at least when we try to mimic as closely as possible natural extracellular vesicles. Although multi-compartment systems can also be very interesting in terms of their therapeutic potential and applicability.
Next question, or set of questions here. Daniele Darigo, can you unmute?
Yeah, thank you. Very great work. And my question is quite more technical. And I would like to ask you if you can control the size of these synthetic EVs and in how much precise you can be. And if it is the case, if you are able to produce a preparation containing different size EV subpopulation within the preparation.
Thank you very much for that question. So I introduced two ways of producing these, either by a microfluidic approach or by a very, standard lab equipment approach, which is shaking these things in an Eppendorf tube. Obviously, the shaking approach will generate a very broadly distributed water and oil droplet population, and that will be represented in your final extracellular vesicle population. It is something that you might want to achieve. Diversity is important for extracellular vesicle signaling, as it is important for the whole biology itself. But maybe in a therapeutic spectrum for regulatory agencies, you might want to prove that you can actually control this in a very precise range. So just to complete this, the microfluidic approach obviously allows to produce a very narrowly defined subset of vesicles at the end. And that is essentially, if you're looking for vesicles in the micrometer size range, very precise in terms of some 10th of nanometer. If you go down to extracellular vesicles, to vesicle sizes that are more relevant for EVs, so below a micrometer down to maybe 100, 200 nanometers, the distribution gets a little bit more broader, but it can be produced in that size range eventually.
Next is Metka Lanasi.
Oh, hi. So I thought that you're just going to read it, but yeah.
No, I'm going to make you talk.
Yeah, I see. Really interesting work, and I think you kind of already answered my question if this is a double layer membrane or it's obviously just unilamellar vesicle. And maybe a follow-up question, how do you think this affects then interaction of the synthetic vesicle with the membrane? Let's say, how is the uptake? Do they use the same pathways? And how does this affect the functions? And I was also wondering about the zeta potential. Yes.
Okay, maybe let me start with the zeta potential. That's the easiest. So as we kind of mimic the lipid composition and protein composition of the extracellular or extravesicular part, that is in the negative zeta potential range. So in between, well, we can be quite flexible in that term, but I think the ones that we report in the paper are around -20 millivolt zeta potential. So that is quite relevant for the extracellular vesicle biology. In terms of how do they exactly interact with cells, that is a very complex problem. And we've been working on the interaction of synthetic vesicles with cells, as has a large community in the drug delivery field done. We have a paper now in revision that hopefully will come up soon that specifies a little bit on a quantitative way and mechanistic way how proteins in such a vesicle will interact and bind with proteins on the plasma membrane of the cell. And I think that's the initial and therefore very important process for EV uptake. And that can be studied rather easily, but anything that happens intracellularly, sorting, release of the microRNAs, honestly speaking, we have no idea about it at that stage. But I think this is again where fully synthetic extracellular vesicles have a potential, because we can at least now start to pinpoint which are the important protein constituents for those processes.
Thank you. It might also be that the functions that you're able to study are more just protein-protein interaction on the surface and then signaling inside. So probably there are different ways, but yeah, very interesting. Thank you.
Thank you very much. Good
points. Thanks, Metka. And in fact, we have a question here that's along the same line. So why are you calling these things synthetic EVs? This questioner Dalir asks, aren't they just liposomes? Why don't you call them liposomes?
I think that opens up a pretty broad discussion about what is synthetic and what is not synthetic. Where does life start? Where does it not start at the end? Essentially, if you ask me as a biomaterial scientist, I would argue that essentially also cells are just very complex liposomes. They're enclosed by a plasma membrane, and anything in there is just very complex to understand. And somehow this evolved on Earth, most probably at some point. So it's just a very complex liposome. Yeah. Obviously, that is a rather complex point of view when you start to incorporate in your thoughts the point of emerging functions and emerging properties that are simply just more than the sum of its parts. And I think that is something that I showed to you today, that liposomes in the classical drug delivery point of view are structures that are rather simple in terms of their composition, and there are almost no emerging properties in those systems. So that is one of the reasons why we call these things fully synthetic extracellular vesicles. But at the end, we also call them fully synthetic extracellular vesicles because they are built according to an EV blueprint at the end. When we build something that is based on the blueprint of a cell, then we will call it a synthetic cell. If we will build a synthetic virus, which is something that we have been doing lately, and we will call these synthetic virions or viruses or whatever. But I agree that this is a that it's something that might be discussed, and different people might have different point of views on the naming of such a structure. But at the end, this is a rather complex and next generation liposome technology.
Very good. Let's go next then to Rayana Safavi, who has questions about the proteins.
Thank you so much. It's a great talk. Since these extracellular vesicles are really heterogeneous, so we don't know about the ratio of the membrane proteins on the surface of the EVs. How did you decide about this ratio, and how did you then measure this? Indicated interaction in the first.
Thank you very much for that question, because it brings up a very important limitation of the technique itself and of bottom-up synthetic biology itself, which is we can only engineer as good as we know our blueprint. And I think in large parts, we don't have quantitative information on extracellular vesicles themselves. So when we were engineering these structures, we basically looked into literature about what is known on the density of membrane proteins and natural extracellular vesicles. And this is how we decided to engineer it eventually. Importantly, and I think I showed this in one of the last slides, we can tune the density in this case and check what is actually relevant and what not. And that might be helpful to rule out some of the hypotheses that arise with natural extracellular vesicles. Unfortunately, I cannot speak too much in this context now about it, but in the paper I mentioned where we study extracellular vesicle proteins in their interaction with cellular proteins, we'd actually take a very close look at how many EV proteins will you need on a single vesicle in order to induce signaling in the cell, how many you might need to induce uptake and so on. So this is a rather complex problem for an engineer if there is no clear blueprint of how to build it and what is the quantitative information that you need to build it.
So it tells us biologists that we need to do a little bit more work here and help with getting those blueprints together. So
more data, the better, I would say.
Yeah, let's get back into the lab, everyone, after this EV club, and we'll get some more data. So, going next to Wenjen Duan, and feel free to unmute yourself, but I'll ask the question here. How big of a molecule can you put into these things?
Again, excellent question. I think for the EV field, anything that's somewhere above 1 micrometer starts to be not too interesting, because that's the usual size range of ectosomes, exosomes, and microvesicles. But as I said, we can engineer droplets with the size of up to 100 or 200 micrometers. And that's essentially the size range that we can also put into their lumen at the end. So we have a paper out in Small where we actually engineered synthetic organelles and put these organelles into living cells. And in there, we actually put magnetic nanoparticles into the lumen of the vesicles in order to give cells kind of a magnet sensing behavior at the end. So you can incorporate microstructures, micro rods of metal particles in these extracellular vesicles if you want, but you can also incorporate maybe more advanced engineering components like DNA origamis, whole viruses, or just very big protein complexes.
All right, next question, Ken Naruse.
Yeah, thank you for a very interesting presentation. So my question is, have you ever compared your synthetic EVs with the natural EVs in terms of the uptake efficiency and the functionality?
Thank you very much. I think that's a very important question that also came up in the revision process of the paper that I presented to you. Obviously, comparing to natural extracellular vesicles is very important for such technologies, qualitatively, but also quantitatively at the end. So what we did is, in an analytical way, compare to natural extracellular vesicles. So essentially, how much of the RNAs that we load into the fully synthetic extracellular vesicles land up in there, and is there any correlation to the natural EVs in this case? But as soon as you start to quantify, for instance, uptake rates, it's very tricky to do this with natural extracellular vesicles. And that starts by problems with labeling natural extracellular vesicles. It's something that's covered in the guidelines of ISEV, that labeling is not simply adding di-I, di-O, or whatever to an EV population. Significant artifacts can arise from this. This is why we have not put any direct quantitative comparison between natural and synthetic EVs into this paper, but it's something that we're still working on in that department.
Next question from Sandra Borata. And so the question here is on different lipid compositions. So what about the biological effects if you vary the lipids that you're using to build these EVs?
So I didn't specify the lipids. No, I think I did actually, just qualitatively, not quantitatively, the different lipid compositions. There are in the paper specified, obviously. But there are some lipids that are rather important for signaling that might be PS lipids, so serine-based lipids that usually induce very potent inflammatory processes. That might be IP3 coupled lipids and so on. We did not vary these parameters in this study, but we know from other studies that anything that, for instance, would change the charge of such a vesicle would dramatically affect how the vesicle will interact with the cell. That is a physical chemistry based process, obviously, but there is also a biochemical nuance to this effect, which is, for instance, PS lipids at the end. So I think there's room for variation and for optimization here, but in this study, we stayed as close as possible to the natural extracellular vesicle lipid layer composition.
Good. So 2 questions here that were submitted at pretty much the same time. So proving once again that great minds think alike. So we hear from Jean-Viet Bar and Yingchao Meng, who both ask about sizes. So, you were making EVs that were fairly large. I mean, when we think about, how the size distribution of natural EVs looks on a graph, I mean, these are very much at the larger end. So what about making smaller ones? Is there a reason that you chose to use this particular size? Would it be possible to go down to 100 nanometers in diameter, or even much smaller than that?
I will start answering broadly. I think in general, producing vesicles that are smaller than 100 nanometers is something that's technically feasible. The challenge is actually not in producing the vesicles, but in getting RNA components, for instance, into the lumen. And this in a qualitative way, basically saying I have 3 RNA molecules in each vesicle, or 3 plus minus 0.2 or whatever. That is a very, very technically challenging thing to do. So this is one of the reasons why we actually went to that droplet-based approach and sequential assembly approach. And in that case, we are limited by the dimensions of the droplets that we can produce. The droplets can be quite small, so you can go down to 100 nanometers at the end, but it's something that I would not say it's feasible in a high throughput and very narrowly defined size range at the end. So everything that we produced in this study was, I think, bigger than 400 nanometers. There is room to go a little bit lower, maybe something like 300 or 200 nanometers. Actually, I think it was 292 nanometers, if I remember correctly. I would need to look it up at the end. So it is possible to go below that. Although with our technique, that might be not the most feasible to do so. Things change a little bit when you're not so much interested in RNAs anymore, but rather care about the proteins that are coupled on the surface of the vesicles. In that case, it's quite easy to produce, for instance, with extrusion techniques or other sonification techniques, vesicles that are much smaller than this.
Yeah, and actually Geneviève has followed up here and asked about the protein, which you just noted that if you have proteins that you're putting on the surface that are in the membrane, you can just do that with extrusion. But is it that the RNA is the problem because it's RNA, or are we just talking about loading anything into the lumen? I mean, is it going to be the same consideration for protein that would be in the lumen as opposed to associated with membrane?
Yeah. So the problem is there for any kind of a component. RNAs are not particularly difficult to get in there. Actually, compared to proteins, it's rather easy to get an RNA or any kind of a nucleic acid into the lumen of a vesicle. You need to start to think about physical chemical processes there, charge interactions between RNA and the lipid. Size effects obviously play a role at some point, but it's not an intrinsic problem of RNAs in this case.
Great. Let's go to Akshay next. Do you want to go ahead and ask your question here about targeting?
Yes, hi. Great presentation, first of all, very, very interesting. So EVs usually have specific targets, and how did you go about getting the correct surface proteins on your synthetic EVs, the identification as well as the attachment?
Yeah, thank you very much for that question. I guess there are different views whether EVs have particular targets or not. Some certainly have, but I don't know if that's is actually verified for all kind of EV subsets. But to answer the question, I think when doing such a bottom-up approach, you can be at a certain point rather ignorant to some facts. For instance, to the proteins that we were using. In this case, because these are tetraspinene proteins, we made sure that the tetraspinene we use are actually in a correct conformation. So the disulfide bonds and so on are there, and the recombinant production systems are the ones that we actually will need. But in this case, we did not specifically select for isotypes of the tetraspinene, for instance. So we did not match tetraspinene producing cells and DNA constructs with the target cells that we're actually targeting with these structures. And that is mostly, again, because we actually don't know which are the correct tetraspanenes that we're looking for in this case. But it's, again, something that could be systematically studied, yeah, with this technology. How do different isotypes impact an interaction between a vesicle and a cell?
Yes, I agree. Thanks.
Thank you very much.
Thanks for the question. And let's go next then to Nasser Jafari.
Thanks for your research. I have a question about the reverse function of the synthetic EVs. Have you tried EVs with different contents that can suppress cell motility, invasion, or wound healing, I would say?
Not so far, actually. We looked also in different RNAs, and there are some processes also in wound healing that you want to tune down at some point. And EVs certainly play a role in such processes. But so far, because of the disease model that we looked at, we were rather focused on, getting things speeded up and into work rather than damping them down, because that's one of the major problems in wound healing therapy. When it comes to inflammatory diseases, the contrary would be the thing probably.
And now a question from Sina Ajit. Can these particles cross the blood-brain barrier?
That's a good question. Honestly speaking, I don't know. Yeah, we didn't test. Yeah, I think I could hypothesize a lot here. We did an initial test with nasal delivery in mice to track if we would find some of these vesicles in the brain of the cells. We ended up in some technical problems of fluorescence detection and so on. But it's something that would need to be tested in an in vivo model, and something I could speculate now a lot based on the in vitro data, but honestly speaking, I don't know.
Good. Well, that leaves us then with some questions that need to be addressed. So we've heard that, the more detailed the blueprint, the better the engineers can design the synthetic EVs. And we also know that once these EVs are produced, there are a lot of questions that we can ask with them. So Oskar, thank you so much for sharing your work today with us. Thanks also to everybody who joined today and who asked questions and interacted with our guests. So it's been a pleasure to host today. I've learned a lot, and I think that a lot of others have as well. So thanks again to everyone. I hope you have a good rest of the week, and I will look forward to seeing you at another EV Club coming up. Take care.
Thank you, everyone. Stay safe.